Hello everyone, my name is Annia Gonzalez. I am a PhD student in Biological Science at the Pontificia Universidad Católica de Chile, and the title of my work is Molecular Analysis of the Circadian Clock Genes in the phylum of cyanobacteria. Circadian clocks are important to ensure better fitness for many organisms. The most ancient clock was found in cyanobacteria, being the simplest organisms that show rhythmicity. Our goal is to understand the evolution of the circadian clock genes among the cyanobacteria phyla. To explore the presence and evolution of the genes involved in the circadian network, we work with the rexed genomes of cyanobacteria. The genomes were downloaded from the NCBI assembly database. To focus the analysis on medium to high quality genomes, the sets were evaluated by check-M software to remove genomes with less than 80% integrity and more than 4 contamination. The final set of rexed consists of 868 genomes. Phylogenomic reconstruction, also known as species trees, was then performed for this set. The alignment was performed using the Marcial software. Therefore, species trees were generated using the IQ-tree software. Node collapse and routing for all phylogenetic reconstruction were managed on the ITOR web server. The list of the 17 genes related to the circadian oscillator was obtained from smelling, and they were classified according to their main role: central oscillator, input, or output. Synagococcus elongatus sequences were recorded, and a blast PCR was performed against nostoc to record homologous sequences and use them as reference for further analysis. In addition, interpersonal software were used to serve this reference for shared exclusive domain. To determine the sequences corresponding to the 17 genes in all the rexed genomes, 3 control points were used when necessary: sequences similarity, present on specific domains, and monophyletic clustering. First, blast PCRs of the 2 reference sequences for each gene was performed against the complete set of predict proteins. The pair sequence were then subject to analysis by the interpersonal software. Domains that were not present in the 2 references were also determined and used to exclude proteins with some similarity and common domains. The alignment of select sequences were performed with the Marcial software using predetermined parameters and used to build the hidden marker models. Using the Hammer software, the models were used as search query against rexed database. In Figure 1, we observe the phylogenomic tree of the 17 proteins of the cyanobacteria clock. In the central ring are the central oscillator proteins, followed by the input protein and then the output protein. As seen in Figure 1, and the analysis showed that only 6.2 genomes have at least one copy of the 17 genes, and the 3 genes of the central oscillation are present together in at least 88% of the genomes analyzed. Analyzing the alignments, we observe that several genes have variants with different configuration based on the present action of domain. As can see in Figure 2, the loss of its N-terminal domain was observed in Chi I. In Chi B, 2 variants can also be observed due to the presence of an additional region and the N-terminal. The functional domain of the rexed protein are more conserved, except for SIG-A, which has 2 variants depending on the present action of the PSR domain. 14 of the 17 proteins show high conservation in the structure of their functional domains, reflecting the robustness of the circadian mechanisms in the phylum. Then we determine whether there was a relationship between these variants, the cell phenotype, and the environment to clarify the distribution and evolution of the circadian gene. For this, we construct this matrix of correlation. Figure 3 and the correlation values in Table 1 show that there is a high correlation between Ka and SIG-A protein that don't present the PSR domain with filamentous cyanobacteria. On the other hand, it is also observed that the presence of the Chi B variant with its long N-terminal domain is highly correlated with both Ka and SIG-A without the PSR domain and with the heterosis forming filamentous cyanobacteria. Thank you for your attention.